Assalamualaikum and hi everyone from Group 4. Today my group are going to present on topic detection of SARS CoV-2 through rapid test kit, RTK antigen test, lateral flow immunoassay. In this video, we are going to cover the principle, method, advantages as well as the limitation of this detection technique. The principle of RTK. The coronavirus antigen rapid test kit is a lateral flow assay that qualitatively detects the presence of nucleocapsid and protein in the specimens. While most of the antigen tests are designed to directly detect the presence of nucleocapsid and protein instead of the other three proteins that present in the virus, is because the nucleocapsid and protein contain the RNA genome of the virus and predominantly express structural protein. RTK is called as lateral flow assay because the specimen or sample move laterally from sample pad across the natural cellulose membrane toward the absorption pad via capillary action. And it is designed with sandwich immunoassay format, in which it is a method using two antibodies that bind to different sites on the antigen. First, the specimen or sample obtained from the upper respiratory system, usually from the nasal swabs, will be received by the sample pad. The sample pad functions as a filter to aid flow. The sample later will move towards the conjugation pad. Here, in the conjugation pad, the sample encounters antibodies conjugated tags like latex or colloidal gold. The sample will rehydrate or absorb the gold conjugated antibodies and bind with their matching antibodies. If the nucleocapsid protein present in the sample, the colloidal gold label SARS-CoV-2 and protein antibody, this one, will recognize epitope on the target molecule, and the binding between it and coronavirus and protein will occur, thus forming antibody-antigen ABAG complex. The sample will continue to flow until reach the capture zone, which is the test line 1, test line 2, as well as the control line. These three lines contain the capture reagent to display a result. The target complex, which is the antibody-antigen ABAG complex, bind to the immobilized antibodies, producing a visible line on the membrane. The ABAG complex is captured by SARS-CoV-2 and protein antibody. The sample will flow until reach the control zone or the control line, where excess conjugate will bind and produce a second visible line on the membrane. And the last part of the RTK is the absorption pad that help with sample flow and also collect any excess sample. And if the specimen is COVID-19 nucleocapsid protein positive, there will be the appearance of red color band on the test line, which is on this T. And if the specimen is COVID-19 nucleocapsid protein negative, there will be no color band appear on this test line.
The first step to use RTK is twist off the cap of the buffer bottle, carefully dispense all buffer into the extraction tube. The second step is tilt the patient's head back 70 degrees while gently rotating the swab. Insert a swab less than 1 inch into the nostril and then rotate the swab 5 times against the nasal wall. Using the same swab, repeat the collection procedure with the second nostril. Slowly remove the swab from the nostril. After that, insert the swab specimen in the extraction tube. Swirl the swab tip in the buffer fluid inside the extraction tube, pushing into the wall of the extraction tube at least 5 times, and then squeeze out the swab by squeezing the extraction tube with your fingers. Gently squeeze the tube's rigid body, dispense 2 drops of buffer specimen mixture into the specimen on the coronavirus antigen test cassette. Lastly, read the test results between 15 to 20 minutes. After 20 minutes, the result is invalid. Negative results show when the color bands appears on the control line and there is no color band shows up on the test line. A negative result indicates there is no coronavirus antigen in the specimen, or the level of coronavirus antigen is below the detection limit. When the color band appears on the control line and the second color band also shows up on the test line, it shows a positive result. It means there is a presence of COVID-19 antigen in the patient sample. Lastly, the test is invalid when there is no color band appear on the control line. An invalid test result suggests there might be insufficient buffer volume or incorrect operation procedures.
The advantages of RTK. First, it gives faster results compared to molecular tests, in which around 15 to 30 minutes only. Faster treatment can reduce the severity of symptoms and also prevent from the spread of the disease. Second, it's very easy to use. RTK can be performed by non-medical workers or volunteers who have been trained to do the test before. Patient may even be able to perform rapid tests on themselves as it don't require specialized laboratory technique. Third, it is inexpensive. RTK is way cheaper compared to RT-PCR tests, thus allowing their use at a large scale. Fourth, no special equipment required. It is useful for use in areas with limited resources or access to specialized laboratories.
There are 4 limitations of RTK. The first one is manufacturing quality and regulatory status. Tests should be procured from manufacturers who work under a quality management system or at least local regulatory approval or right of free sales granted by the country of manufacture. RDT, as all in vitro diagnostic intended for clinical use, should undergo a rigorous and transparent regulatory review. Next, shipping and storage conditions and shelf life. The capacity to withstand temperature stress and having an extended shelf life are critical to the ease of use of AG-RDT. With new product, shelf life must be estimated based on accelerated stability studies, but target shelf life should be at least 12 to 18 months at 30 to 40 degrees. A cold chain requirement for shipping or storage would significantly increase the cost. Besides, the cost of the test is also one of the limitations. The cost of tests will vary according to the test and the volume to be purchased. In general, they should be less expensive than PCR tests. The cost of transportation, import tariff, storage, and user training, and post-purchase quality control testing activities required to support quality implementation of RDT must also be considered. Lastly, test procedure. Failure to follow the instruction for test procedure and interpretation of test result may adversely affect test performance and produce invalid result. That's all from our group. Thank you.